Hi, my name is Shantanu Shukla and I'm a PhD student in the Genome Sciences and Technology program at the University of Tennessee, Knoxville. I'm working in the lab of Dr. Dean Miles at Spallation Neutron Source and Oak Ridge National Laboratory, where our lab focuses on understanding the structure of biological macromolecules, especially proteins, using the techniques of X-ray and neutron crystallography and scattering. My research currently deals with understanding the structure of bacterial ATP binding cassette transporters, or simply the ABC transporter systems, that help not only bring in the nutrients inside the cell, but also to flush out harmful chemicals such as drugs and antibiotics outside of the bacterial cell, which otherwise might be toxic for them. But determining the structures of biological macromolecules is not a very trivial task. They often require a lot of expertise because they are very tiny, and even for a very high resolution microscopes, they're sometimes difficult to understand. And so that's why we use X-rays and neutrons to peer deeper into the atomistic details of these complexes. So far, I've used the techniques of X-ray and neutron crystallography and scattering to understand how these ABC transporter proteins can specifically pick a certain substrate molecule from a larger pool of chemicals in the bacterial periplasmic space. Now, the periplasmic space is an aqueous environment between the bacterial outer and the inner membranes, which acts like a nutrient pool for bacteria. These ABC transporter proteins have evolutionarily been conserved in terms of their structure, but they are very specific to the kind of nutrient that they bind to. For example, a sugar binding protein will never bind a vitamin and transport it across the membrane. Therefore, the specificity is very important for these systems. Also, these proteins have a different dynamics, that is how they move in the solution or in the cellular environment. So that dynamics again offers them this specificity towards a certain kind of substrate molecule. Finally, understanding the ABC transporter system will not just allow us to design better microbes for industrial use or biotechnological use, but can also help us understand how bacteria gets resistance to certain drug molecules and then help us to design therapeutic interventions to cure bacterial drug resistance.